17 years ago, I was 14, attending Connor Middle School in Hebron, Kentucky, a small little town just across the Ohio River from Cincinnati, which, by the way, is where I was born, though I spent most of my life in and around Houston, Texas, or Minnesota. In 2018, I moved away, back down to Houston, but in the few years that I was there, I met and became friends with some of the most amazing people. But, as life goes, I lost contact with almost everyone over the years. Then one day, sometime last year or so, a familiar name popped up on my feed. An old friend, and today's guest.
My name is Tiffany Slater, and my pronouns are she, her, and I'm based in County Cork in Ireland, and I work as a researcher at University College Cork.
Dr. Tiffany Slater. She's a paleontologist now, taphonomist more specifically, but we'll talk about that more later. Studying ancient biomolecules and absolutely killing it alongside her team and making some brilliant discoveries in recent years. Today's episode, searching for molecular ghost in the fossil record, ginger frogs, ancient proteins, learning there's a difference between aging fossils and dating fossils, feathered connections, archaeopteryx, perspectives, particle accelerators, and of course, catching up. I'm Devin Boker. You're listening to The Wildlife. So, journey, because I know you from way back, and split off at some point, and so how what was your whole journey getting to where you are now in Ireland?
Oh, I often have to remind myself how I got where I am today. Because life has been a crazy, unplanned, beautiful thing so far. I don't know if you had a similar experience to me, but growing up in Kentucky, I had kind of a very poor understanding of evolution, and I was never very good at science, so I was always very insecure about sciences, and never had any interest becoming a scientist. When we learned about evolution in science class, it was always evolution on a micro scale. So it was about, maybe how bacteria would evolve, or, how, maybe the molecular process would evolve and change. But, we would always kind of deal with timescales that were very short, and we never really got into the topics of macroevolution. And I also grew up as a young Earth creationist, so I never really questioned when we were kind of taught about evolution on a micro scale and not a macro scale. It kind of suited my view of the Earth. So I, really, it was how I got from where I am today. How I got to where I am today is mostly out of depravity, I'd say, because I went to university and I took a biology course as a gen ed, and I definitely didn't want to take the course. It was just because it was required that I did. So I started learning about macroevolution more and Darwin's finches, and it just captivated me. I had never heard about it, so I think it was kind of that, that when you see a baby and they get so excited because they see something new for the first time. It was just like that for me. I was just captivated by how fascinating evolution was. And so at that point, I was studying sociology and history and anthropology. So I was in the social sciences, and I still find them really fascinating today, just how people work and all that. But I was so fascinated by macroevolution that I just went ahead and changed my major. So I started studying biology, and ended up researching, helping in a research group. I was volunteering my time, in a group that studied blind Mexican cavefish.
Also known as the Mexican tetra. There's actually several different types living in different areas, parts of Texas, the Rio Grande, parts of Mexico. Sometimes people consider the blind cave form a separate species, but I think that's a subject of debate. Super interesting little fish. And you know what? I'll pop a link in the episode notes in case you want to do further reading about them.
And they were looking at how the fish ended up losing their eyesight and their eyes, and how the bones
Yeah.
changed around their eyes. And then I kind of knew that research was an area that I'd like to go down. And then long story short, ended up moving to the UK to start fresh my degree at a university called the University of Worcester. And I finished my degree there. So it took me 3 years to get my undergraduate degree in biology, which seemed very achievable for me at the time because I had been studying social sciences for such a long time. So so I studied there, and then I think it was just my naivety of being in a new place and not knowing the politics, and not knowing who I should or shouldn't contact. I ended up contacting a professor of paleontology at the University of Bristol, who is a renowned paleontologist. And I emailed him asking for some voluntary work over a summer. And lo and behold, he got back to me. And then I had asked for his textbook for Christmas. My mom bought me his textbook for Christmas. And then I looked down and I saw his name on my textbook.
Professor Michael Benton. He really is a big deal. He works on large-scale evolution of major groups like dinosaurs, and he explores the effects of mass extinctions and environmental change and biological innovation on the evolution of reptiles, birds, and mammals. And you want to talk about someone who has published a lot? He has been an author or co-author on 488 articles in academic journals, 67 books, 63 chapters in different books, and like over 30 other things as far as I can tell.
And I was just like, oh my gosh, I've been emailing this man asking him for voluntary work, and had no clue, how kind of prestigious he was and how intelligent this man was. But I ended up doing an internship with him at the University of Bristol. So I volunteered for 4 months and ended up publishing a paper with him out of the work that I did there. And honestly, that was kind of the start to where I am now. It really set me up, and I was able to get a PhD position here in Ireland. And so I finished up my PhD last year. I graduated, so I'm a postdoctoral researcher now. So yeah, that's in a nutshell, a long nutshell. That's how I am where I am.
No, yeah. I mean, there's a lot of that I can relate to, because I mean, yeah. Similar kind of growing experience and stuff. And, I hadn't actually made that connection or really thought about that until you just said that like, the way that we kind of learned about things was kind of on this micro scale. And then I think about it, I'm like, yeah, we would talk about like E. coli in particular, like a lot about E. coli and stuff. But I don't remember. Either I wasn't paying attention because I was goofing off, or I genuinely did not have the experience. I don't remember learning about a lot of this stuff on like the macro.
Yeah.
And yeah, I mean, at my home, that was kind of like, we went to church every Sunday, and had that view. I remember, and this is sometimes funny for me to think about. I remember like being on YouTube. It was new and stuff like back in the day. And I remember like arguing with people about like evolution and how it didn't make any sense. I remember doing that. And let's take a quick sec to define evolution, because I feel like all of us really innately understand what it is. We see it on a daily basis. We are a part of it. And yet so many of us reject it because of misconceptions. Honestly, I don't even like that word, preconceptions. You know that you get half of your DNA from your mom and half from your dad. You are a shuffling of their genetic cards. If you have children, you will pass on half of your genetic information with the person that you have the children with, therefore shuffling those genetic cards even further. Generation to generation, those genetic card decks are shuffled over and over and over again. Sometimes traits that were seen in the grandparents pop up again. Sometimes they disappear entirely. Even siblings with the same parents can have different traits. It all depends on which genes are present. Now, that's a family. You scale that up to a population level that change in the proportion of genes over time. Which genes are there, which aren't, which are more common, which are least common. That is evolution. Maybe you have a set of genes that makes it more likely that you will live long enough to have children and pass on those genes. Well, congrats. You have just achieved reproductive fitness. You have just ensured that those genes are going to continue forward into the future. But maybe you have something that makes it less likely that you are going to live long enough to have children. Or maybe you just don't. Those genes, they don't get passed on. That is the core idea of natural selection. Which, by the way, and since we're here, I said reproductive fitness. That is the true idea behind survival of the fittest. Survival of the fittest does not mean the strongest, the meanest, the fiercest, the most violent. That's a bunch of alpha silliness. Survival of the fittest means what was passed on. Which genes persist. You know, now that I think about it, I should probably do a whole episode on like common misconceptions about evolution, but now's not that time. Let's get back to it. And then here I am now. I've got the Darwin finches tattooed on my forearm.
Do you actually? Oh, that
Yeah,
is
you mentioned the Darwin finches.
cool.
I was like, oh. And I'm probably getting another I'm getting another Galapagos themed one on Saturday.
Oh,
Actually. It's
stunning.
my students, I had made a deal with them that if they all got to a certain point that they could submit designs for my next science themed tattoo. And sure enough, they got to that point. And
Of
so
course
now I
they
have to
did.
hold, so now I have to hold to my word. Fortunately, it's not that bad. It's silly, but it's not that bad. It'll just be on me
Oh,
permanently.
what an incentive.
Yeah. But it's so the inspiration is Galapagos. So it's like your classic ghost, but it's the outline of a saddleback tortoise. And so so it's a Galapagos tortoise, and I'm like, okay, that's honestly kind of funny, and
Dear,
I'm okay with it.
I might need them to design my next tattoo.
I was kind of shocked. I was like, there's no way you were that creative, but okay. No, but yeah. Life, yeah, you just kind of like academics in particular, it's kind of just ends up being like this cascading effect sort of thing. You know, you work in one thing and then that takes you down this path, and then you meet somebody and then that takes you down another path. And but that's just
And
so
life
interesting.
is so beautiful that way. Like,
Yeah.
and I did kind of feel like it's funny. Just speaking of tattoos, I have a skull of archaeopteryx on my arm with
Oh,
the
that's so cool.
feathers that were associated with the archaeopteryx fossil. And so I do, I got that long before I started working on fossil feathers. It just kind of shows you, it's just like you just start doing things that are true to yourself, and it things just happen.
Advice. I know this is always a hard part, especially because life is so unpredictable.
Mm.
But if you could, even if it's just to your younger self, what advice might you give someone for going down that path?
Yeah, like going down my path in particular,
Or sure,
just life.
that or
Yeah.
the sciences, or
Yeah.
yeah.
Yeah, I guess well, I suppose that would be my advice really, is to not try to go down any path in particular. Like, I mean, of course that's great if you do know what you want to do, but I think I would go back and reassure my former self that it was fine if I didn't know what I wanted to do. Because sometimes life can be more beautiful than it would be if you had just planned it all. Because there's things in store that that might not exist, things that might not exist right now that will exist when time is right for you to go out and choose a career or, make a decision like that. So I'd say instead of worrying about making some sort of 5-year plan or something, that just get curious about the things around you and spend your time reading about the things that you enjoy the most, and maybe just trust yourself that opportunities will come and find you curious and working.
I think that's wonderfully said. Especially, yeah, the curiosity piece. I think that's like, I wouldn't say missing with a lot of people per se. Like, I don't want to say missing, but I think, yeah, sometimes people get so caught up on the plan that they
Yeah.
miss the things that are right in front of them, and like they limit their ability to chase their curiosity
Yeah, I
for
think
things.
curiosity is not encouraged maybe as much as it could be.
Yeah.
But like, if I'm thinking to, we took so many tests in high school about what kind of job are you going to have when you get older. And I
Hmm.
think that mine actually told me that I should be a trash collector. And like, there's no shame in collecting trash, and
Yeah.
I'm sure that they get paid better than I do, and have better benefits. But I think that was maybe more so picking up on maybe my compulsive tendencies to keep things tidy rather than what I really wanted to do in my heart.
Yeah. Yeah. Mine. What did mine always say? Mine always said dancer, and I'm like, you
Wow.
have not met me. I don't dance. I
Yeah. Yeah.
I,
It's,
I can't. I can keep rhythm, and I can keep a beat, and I can do that with like music. I cannot. There's a disconnect from here downward. I just can't do that. So I don't know what you're on about.
yeah, see,
Um,
the logical thinking for your career progression is not it's not the
yeah,
best way to go.
it didn't make any sense. I'm like, that is not going to be something that I fall into at any point. I can't imagine. Um, okay, so your current stuff. I was reading the things that you sent over. Absolutely fascinating. How did you? Because you mentioned the Mexican cave fish, and then the paleontology. Um, how did you get into things specifically related to like birds and feathers and that stuff?
Yeah. So when I was interning at the University of Bristol, I was really interested in fish. And so I am a bit surprised to have ended up working on birds. But again, it's just about being curious, really. And um, what I was doing working on fish, it all kind of ended up relating to each other. So I was, after Bristol, I went and I did another internship at the University of Vienna, and I was studying a fossil shark. And fossil sharks they're really interesting because normally sharks would just preserve in the fossil record as their teeth. Like you would
Hmm.
just find isolated random bits of teeth.
The reason being that most of a shark skeleton is cartilage, which is considered soft tissue and doesn't exactly preserve very well.
Sharks have that conveyor belt where they just constantly producing more teeth, and those just get, you can find loads of them in the fossil record. But I was actually working on one fossil shark, an entire body of this fossil shark. It was a baby shark, so it was about like the length of my forearm, and it um it had preserved the outline of the body, and it was just so fascinating because it was the type of fossil that I hadn't really worked with before, and so it ended up leading me to my PhD, which was working on soft tissue preservation in the fossil record. So you get so many bony things in the fossil record
Yeah.
and bits of broken shell. That's like 90% of what the fossil record is composed of, is broken bony bits and broken shells.
With that, I do think maybe it's important to pause for a moment and talk about how fossils form. There are technically several different ways, but most of them are formed when a living organism, like a plant or an animal, dies and then is quickly buried by some kind of sediment. Maybe that's volcanic ash, Pompeii, or mud, or sand, or silt, something. The soft tissues are going to decompose. I mean, sometimes they can persist, but it's got to be really special circumstances for that to happen. Part of the importance of being quickly covered is because it slows down the decomposition and, prevents things like scavengers, other animals from getting at the body, pulling pieces in other places, and it just kind of keeps everything more or less intact, the way that it was when the organism itself died. Now, to throw another technically at you, there can be body fossils and trace fossils. Trace fossils, they might be something like coprolite, which is poop, or footprints, or burrows, or nest sites. Body fossils are, of course, parts of the body. As far as the body fossils go, sometimes it is the original parts that are preserved. Sometimes it's almost like a cast. Like, for example, the process of permineralization, where empty spaces within the cells of the plant or animal begin to fill with sediment carried by groundwater, which eventually crystallize, basically replacing those structures with rock in the shape of the original organism.
And so this shark, it was preserved with soft tissues. And so then I ended up doing my PhD on soft tissue preservation of feathers. So it was kind of everything linked up.
Yeah.
Yeah.
Yeah. That's one thing that's always kind of fascinated me in particular. And like, I'm always so like geeked out when I see like something pop up in the news about like, them actually managing to find like a jelly fossil,
Oh,
in a riverbed in China or something. And I'm like, that's
yes.
just amazing because like, they're nowhere. They're like
Yes.
things like that, just you don't find them. And so
No.
finding things like that is just fascinating. We'll get to, I guess, the more specifics and stuff of the ones that you sent over in a minute here. But just as a couple general things, like, because this feels like it's within the same realm. Something people wonder, right? Like how do we look in the fossil record and interpret things out of the fossil record? One of those being color. And so
Mm.
like, there's been a lot of change, you just even dinosaur wise, right? Like in the 90s, how they were depicted in pop culture to today, how they're depicted in pop culture when, suddenly there's feathers and colors and like how do we know the colors of things in fossils?
Yeah, it's such a good question. I think, if you'd asked people 20 years ago, they wouldn't believe that we would have any idea today what the colors of dinosaurs were. But the research in fossil color started about 15 years ago. And that was when a paleontologist was looking at a fossil feather and under a scanning electron microscope. So it allows us to see the most minuscule of things down to the micron scale. So we have in our hair and in our skin, we have melanin pigments. And this is what makes your hair brown or ginger colored, or it's also what's in your freckles. So these pigments are stored in these little organelles called melanosomes, and they are about around 2 microns in size. So they are, you can't see them with a normal microscope, but you can with a scanning electron microscope. So it, throws loads of electrons, which are super, super tiny, at the fossil and then creates an image for us. So that's how we're able to see that these melanosome organelles, they can preserve in the fossil record. So we find evidence of melanin pigments in fossils, and that's how we're able to start to determine the original colors of the dinosaurs.
So are there different kinds of is there a difference in the melanin, like the structure or type of melanin that might indicate different kinds of colors?
Yeah, exactly.
Okay.
So now this is definitely going to oversimplify matters, but broadly, there are two types of melanin. So we have eumelanin, which makes black and brown hues. And then we have pheomelanin, which produces more gingery, rufusy red, and gray coloration. So these two pigments are packaged in melanosomes that kind of differ in shape. So in
Okay.
a lot of tissues, your pheomelanosomes that contain the ginger pigment, they would be like a ball shaped.
Hmm.
And then your eumelanin inside eumelanosomes are more like sausage shaped. So, and this is particularly true of feathers. So, this kind of binary state of the melanin. So in fossil feathers, we're a little bit more easily uh, able to tell what kind of melanin color at least
Hmm.
was in the tissues.
Okay. Are we able to tell other colors as well? Like, I mean, I can't even remember how, like, cardinals get their color from food. I'm not entirely sure how that pigment exists within the feathers and things. Or can we see other colors outside of the like melanin based ones?
Yeah, so this is definitely something that paleontologists are working towards. So, because melanin makes up a lot of the color generated in vertebrates,
Hmm.
but it's not everything. We know, as you said, there's other pigments. There's carotenoids, which can generate this sort of vibrant greens. There's xanthophores. So there's loads of other types of pigmentation that we can find in modern animals. So we do suspect that those would have originated at some point. So we do suspect that they could be in the fossil record, but we have yet to find any evidence of them.
Sure.
Um, so right now, that is a kind of caveat to some of the reconstructions of the original coloration of
Hmm.
extinct dinosaurs, is that these are melanin based reconstructions. So not including some of these other secondary pigments that could be involved. And that's even before we talk about structural color. So
Yeah. I guess so, like there's some level of like inference, I suppose, that could be done where, you're looking at existing organisms and you're looking at the environment and the coloration of the like there's matching and things that happens between like organismal coloration and the environment that they live in that kind of makes sense. It's not always cut and dry. For example, a cardinal, is very, very vibrant and stands out. And so, like, your typical idea of, well, maybe it's blending into the environment doesn't exactly hold as much. But I would imagine that it can be useful in trying to put these things back together.
yeah, it's possible. Well, and something else that's interesting to do with the how the environment kind of shapes your color is
Mm-hmm.
that there's evidence that shows that the counter shading on an animal relates to the habitat that it's living in.
Mm-hmm.
So, and this is probably the easiest example is a deer. So you can see the white underneath side of the deer is very clear, and it's in stark contrast to the darker part of the upper body of the deer that would be exposed to the sun.
Mm-hmm.
So when that line between the white and the dark is higher up on the body, then it's more likely to be living in an habitat. So, one that's kind of clear from
Hmm.
vegetation and that's so that, when predators are seeing this animal on the horizon, that it's more blending into its habitat, um, because its line of color is more kind of along the middle of the body. And then let's say this animal was living in a closed habitat, so like in a forested area, this the line between the dark and the light part of the body, this counter shading area is going to be more likely to be lower on the body, so more type around the belly area.
This idea of counter shading, you can see it everywhere in nature. And you can even see variations of counter shading with spots. Think a baby deer. Those white spots along its back mimic the way that light pierces through the tops of the trees. So not only do you have this sort of, break in the horizon based counter shading, but you have counter shading based on the way that light plays with the environment. Heck, you see this in a lot of oceanic organisms too, with swirls and spots on their top, so that predators from above might just think it's the light hitting the sand at the bottom. And it's not just prey animals. There are plenty of predatory animals that take advantage of this kind of coloration as well. How in the world do we look at fossils and determine how old a fossil is? And I know some people are just like, oh, it's carbon dating, but there's also more to it than that. And then there's also some like distrust, right? Because people are like, well, how in the world do you know that this thing is actually this old? And this could be from, 20 years ago. And so how exactly do we look at a rock essentially and know how old it is?
Yeah, carbon dating is an important way of telling how old a rock is, but
Mm-hmm.
it's not the only way. And I think the perhaps the easiest way is to think about it like a cake. So, when sediments are being deposited by a river or on land, it's these sediments are getting stacked layer and layer and layer and layer, just like a cake. And when you look at a cake, you do presume that the first layer, the bottom layer of the cake, was laid down first. And we read the rock record the exact same way. And then we know that the icing was put on last.
Mm-hmm.
So, when we're looking at rocks, and let's say we extract a fossil out of a rock, we can look at the rocks above it and is kind of work our way backwards and say, okay, well, if these rocks at the top are this age, and we know that, then you can work backwards and you start to be able to read the rock record and go back in time. So that would be the simplest way of determining approximate age for a fossil.
Okay. Well, that actually makes a lot of sense. The Earth is a cake.
Yeah,
With a molten core, so it's a delicious cake.
exactly.
That makes more sense. So you mentioned, and then you mentioned the U melanin. Did I say that right? The U melanin, is that the one with the ginger that produces ginger,
The
or is it
ginger
the
is actually
theomelanin?
produced by pheomelanin. Yes. Yeah.
There we go. Okay. All right. So you had sent over the bit about finding the ginger pigment in, what was it, 10 million year old frog
Yes.
fossils, which is just so cool. And also so cool that frogs have been around for as long as they have, because like, come on, frogs are amazing. It's composed of, and I feel like I'm just saying it wrong. Say it again.
Is
Which
it
kind of melanin?
pheomelanin?
Pheomelanin.
Uh-huh.
Okay, pheomelanin. So that's what it's composed of. What does this discovery mean for those who, sometimes people are kind of picky about science and they're like, well, what does that even matter? What's the point? What can we learn further from this, and what's the significance here?
So there's a lot of different answers to that question. I kind of love hate getting that question. Um, so to kind of hark back to what I was saying about the habitat
Mm-hmm.
interpretation, um, due to the color of an animal, we have been able to interpret the habitat that certain fossil organisms would have occupied. So, due to studying their color patterns. So, for example, we know that um, the dinosaur called Sinornithosaurus. So this dinosaur would have lived in the early Cretaceous, and because of where that counter shading line is on the fossil dinosaur, we know that it lived in an open habitat. So, and similarly, there's an ornithischian dinosaur called Psittacosaurus, and because of where its counter shading line lies, we know that it would have lived in a closed habitat. So, you know, finding evidence of pigmentation in the fossil record can tell us a lot more about extinct animals than we would have known previously.
Mm-hmm.
Um, and, all of that helps us interpret and understand the world around us and understand how that has changed through time or how it has been conserved through time.
Yeah.
So that's just to kind of reference back what I was saying there. But so yeah, that's a nice example that we can actually start to understand animal behavior through
Yeah.
the fossil record. It's all about how these animals were interacting with their environment or interacting with predators or through camouflage or even mate signaling. So Sinornithosaurus also has this striped tail. And we know that because there's evidence of these melanosomes packed in striped layers
Oh,
all along
sure.
this really tall tail. So this is interpreted to be a kind of way to signal to other members of its species that it's that it is one of them. So fossil color can tell us a lot about animal behavior and the habitats that these animals lived in. But I suppose, thinking about it a little bit more broadly, we don't have a great understanding of how these pigments evolved and when or how they originated. By finding evidence of pheomelanin in the fossil record, it does start to allow us to build a better understanding of how and why this pigment evolved.
Yeah.
And that's something that we're still working on to try to answer.
Yeah.
But I suppose an even broader answer to your question is probably just that we all need a basic understanding of science to navigate life. And I think a good example of this might be COVID. But the thing is that paleontology and findings like this can capture people's imaginations. And it's really a gateway to get people interested in engaging with science or maybe even pursuing them to become a scientist. It's important that people are able to understand the basics of science so that they can understand the news and understand how things are affecting their daily lives.
I'm going to play that for my students. That's because they hear from me and they're like, okay, okay, okay. The more they can hear that from other people, the better.
Yeah,
Because I'm always on that. Like, come on, it's not just about here. It's not.
it's
It's about
not.
way more than that. It's
Yeah,
bigger.
it's also relevant because so we understand that eumelanin, the pigment that produces black and brown coloration, we understand that that's really important to animals because it protects our tissues from radiation damage from the sun. And not only that, it can make our tissues stronger. So when you see black on the ends of feathers, for example, that can help protect their feathers from just mechanical abrasion from flying around in a tree. And, a
Hmm.
lot of times if you're walking around, you can hear the pigeons flapping and the crows flapping their wings in the trees. And it's really
Yeah.
loud. And, that's because they're hitting those branches really hard. So that eumelanin pigment can actually strengthen tissues like feathers. But the thing is that pheomelanin is a bit different. We don't fully understand its purpose. So, eumelanin is toxic to produce, but
Hmm.
it has very important properties that can help protect our tissues from damage later. So, there's a bit of a trade-off there. It's
Yeah.
costly to produce, but then it has a benefit in the tissues. But the thing is about pheomelanin is that we don't really understand why it's of benefit to our tissues. So, we don't really understand why ginger pigments in our skin or our hair might protect us. It doesn't seem to have a protective function.
Hmm.
So we don't understand why it evolved and how it evolved. Um, and this is something that's implicated in a lot of different things. We know that um, uh, people with ginger hair and are more likely to get things like melanomas, and it can become really relevant if we start to think about it in a biomedical perspective. Um, and the fact that we don't know how or why it evolved. So hopefully this finding evidence of pheomelanin further back in time will give us more tools to be able to detect it in more fossils and
Yeah.
especially older fossils to try and help answer this question of how and why did pheomelanin evolve in the first place.
So on a sort of similar note, the other thing that you had sent over was equally as fascinating, but it touched on there's not just it's not just melanosomes and pigments and things that you can find within the fossil record. There are other, I believe, smaller things that you can find, which are equally as fascinating, proteins. So tell me a little bit about that, because it was kind of just when I was reading it.
Yeah, well, see, and this is also something because paleontology historically is kind of something that people associate with old men, finding bones and stuff
Yeah.
like that, and
Yep.
really, um, I do love how modern paleontology is just really where it's really pushing the boundary of what we can get from the fossil record and capture people's imaginations even more. So there's a lot of debate about how long proteins can survive in the fossil record. And it's understood that most proteins can only survive for about 10,000 years before they degrade and there's, you know, almost nothing left.
Hmm.
So it's it's quite controversial that a protein might survive for a million years tops. Now, the oldest evidence of proteins in the fossil record at the moment comes from a camel that is about 3 million years old, and it's from the Arctic tundra.
Now, I know what you're probably thinking. Tiffany did just say camel fossil from the Arctic. Now, at first, that might sound ridiculous, but if you think about it, it's a very similar environment to the desert. Resource poor, fairly empty, have to conserve water. Plus, believe it or not, there were plenty of native camels and horses to North and South America. That is, until people arrived. Oh, the great flood of humanity wiping out most of the megafauna on Earth. That's a topic for another time. Oh, but one more thing. Camels used to be about the size of a rabbit. So just picture that. A camel the size of a rabbit. Mm.
So that's pretty remarkable
Yeah.
in and of itself. It's especially since we think that proteins really only last about 10,000 years, and that's in a very special case. This is evidence of proteins preserved in ice.
Yeah.
This is
Right.
the best case scenario for protein preservation, um, and something that is starting to be looked at more is traces of proteins. So, trying to find evidence of the protein in some way, shape, or form.
Sure.
So, we're no longer expecting to find intact proteins back as far as 100 million years. When I was doing my research in my PhD, we were trying to find evidence of the proteins.
Essentially like the shadow of a protein, right? So then maybe not finding the protein itself, but something that indicates that there was a protein there.
Yeah,
Is that
so
okay?
so unlike we might not be able to expect something like is preserved in that camel from the Arctic. We
Yeah.
might not be able to find proteins as well preserved as that, but there is a chance that we can find evidence of the protein in some
Sure.
way, shape, or form. So, this could be a fragment of the protein, or yeah, it's just some sort of trace of the protein.
Hmm.
So, for my PhD, I was looking for evidence of keratin proteins in the fossil record. So, keratin proteins are what makes up your hair and your skin and your
Hmm.
nails. Um, and we were able to find evidence of traces of these keratin proteins in fossil feathers from a feathered dinosaur called Sinornithosaurus, and which is about 125 million years old. And it was found in China from a place called the Jehol biota. And there, we also found evidence of these proteins in a very early bird called Confucius ornis, which is also it's from a similar locality to Sinornithosaurus. It's about 125 million years old. So
Mm.
yeah, so it was really, really exciting because it does push back this idea of how long proteins can survive in the fossil record, if not intact, at least through some sort of fragments that we can then interpret and gain more information from. The biochemistry of modern bird feathers is really unique. And that's because the corneous beta proteins that they're composed of, it's they're really, really durable proteins. And the reason why there's part of the reason why they're so durable is because they have so many disulfide bonds. And this bonding is particularly strong. So when feathers are degrading, this it really it halts the process of decay for feathers. So it's probably why you go around and you see so many pigeon feathers on the ground, and they're so hard to degrade. So the poultry industry ends up amassing tons and tons of waste feathers every year because
Hmm.
we eat the chicken meat and then they don't know what to do with the feathers.
Yeah.
So there's a lot of research going into how to degrade these feathers. And it's because these corneous beta proteins are just so tough. So these disulfides are really special. And this might be something that, that's also found in some other proteins, but there's just so many of them in feathers. And there's very little else in feathers other than corneous beta proteins and melanin pigments.
Hmm.
So it means that they're relatively, let's call them quote unquote, simple structures biochemically. So when we're looking for traces of corneous beta proteins in fossil feathers, we thought, why not look for disulfides? Because we know that these corneous beta proteins
Oh.
have so many disulfide bonds, and that's what's making them strong. And if that is the strongest bond that is within the corneous beta proteins, then we might be able to find them in fossils that are hundreds of millions of years old.
Yeah.
Luckily, we did find disulfides. So this was one way that we looked for traces of corneous beta proteins.
Hmm.
And then another way, actually, in order to look for those disulfides, we ended up having to take our fossil feathers to a particle accelerator in
Wow.
Stanford, California. So that's really exciting because you're in this massive building with wires everywhere and radiation signs and danger warnings and loads of red buttons that you just want to push. and it's a really exciting place to work. But we had to take them to the particle accelerator because these disulfide bonds, the sulfur is present in such tiny quantities that we needed a really powerful light source to generate the energy needed to detect the sulfur.
Hmm.
So that was really exciting. And then another way that we were able to look at traces of corneous beta proteins is we used a piece of equipment called an FTIR, and it carries infrared light inside of it, and this infrared light vibrates bonds, and so every chemical bond has a different way that it interacts with infrared light.
Huh.
So there are certain bonds in proteins that absorb the infrared light at a certain wavelength. And so we looked for evidence of that wavelength in our infrared data to see if there was any evidence of proteins. So it's the NH bond in any protein. Any protein that you can find will have an NH bond, and this will vibrate at a specific wavelength when infrared light hits it. So we were able to find NH bonds in our fossils. So that tells us that there is evidence of proteins in our fossil feathers. So using these different techniques, we were able to detect the disulfide bonds using the particle accelerator, and then we were able to detect NH bonds along with some other bonds that are indicative of proteins using
Yeah.
infrared light. So you don't need a whole protein to be able to detect it in the fossil record. You're doing chemistry, which
Yeah.
I never thought of myself as a chemist. So but it's remarkable that we can get these bonds preserved in the fossil record.
And so in this particular case as well, it was also there's also some linkage too, right? Between I guess the structure of these dinosaur feathers and like bird feathers are like a more traditional idea of like a bird feather.
Yeah, so it was previously thought that so when I say keratins, really I should specify that keratins have kind of they've been classified into these two different types of keratins, so alpha keratins and beta keratins. I'm
Mm-hmm.
not sure if you would have heard this distinction before, but it's kind of thought that these beta keratins are what makes up the hard keratins, so the stuff in your fingernails, and the alpha keratins was more like the
Okay.
stuff found
Yeah.
in your hairs. We now know that alpha and beta keratins is a little bit misleading because these are actually completely different proteins.
Hmm.
So calling them both keratins is a little bit unfair. So we now know that beta keratins, which is in your nails and in feathers, they're now called corneous beta proteins to better differentiate them from true
Okay.
keratins. So if we refer to them in that way, there was this thought that, so modern birds, when you go outside and you pick up a feather off the ground, that's mostly composed of corneous beta proteins. And it was thought before that feathers had evolved from these simple, more flexible, true keratins, and that they then over time hardened to become more stiff and more durable for sustained flight, that they then evolved and were mostly composed of corneous beta proteins. So, and this is it kind of tallies with the thought of in the evolution of flight and thinking that feathers first evolved for a purpose like insulation or
Mm-hmm.
color signaling, and that they then later were used and co-opted for flight. So now we were looking at Sinornithosaurus and Confuciusornis, this feathered dinosaur and early bird, to see if we could find evidence of corneous beta proteins. And Sinornithosaurus is a very, very ancient feathered dinosaur. So if this hypothesis was true, that feathers went through this evolutionary transition from a keratin to a corneous beta protein state, then we would have expected Sinornithosaurus feathers to
Yeah.
mostly be composed of keratin. But that's not what we found. We actually found that the Sinornithosaurus feathers were dominated by corneous beta proteins.
Hmm.
And this was a little bit of a shock, because well, one, we didn't know whether or not traces of these proteins could survive.
Right.
So, um, so it was really remarkable to even find that fragments of them can survive for 125 million years old. And then on top of that, that we were able to kind of determine that the traces that we did find were more indicative of corneous beta proteins than they were keratins.
Which tells us the biochemistry of modern feathers is a lot more ancient than we thought.
That feathers were, they had evolved quite a lot.
Yeah.
They had undergone a lot of evolution even 125 million years ago. And we actually still don't know when the first true feathers were formed.
Yeah.
It's, you know, it's still an important question that we need to answer is how and when did feathers evolve? And this does kind of push back our our idea of when the biochemistry of modern feathers first evolved. And now we know that 125 million years ago, the feathers were remarkably quite similar to modern day feathers.
That's really cool. Honestly, it's like see paleontology, I mean, well, biology in general, but also this, it's like microbiology through the eons. It's like really fascinating because like you said, like the traditional idea is like you're like assembling skeletons and it's like this big macro stuff, which is kind of contrary to what we were talking about in the beginning about learning about
Yeah,
evolution, right? Where we're talking about, learning micro and not the macro. It's like paleontology has always been this macro, and now we're getting this micro to it.
so
And that's
right.
just amazing.
You're so right. And this is obviously the macroevolution and how organisms can evolve to become a different organism is entirely fascinating. But it's equally fascinating to understand how the molecules that make
mhm.
up our lives today, how and when they first evolved.
Yeah,
Because there's so many things about the natural world that we just still don't know. And a part of
right.
me loves that, and a part of me sees it as a challenge.
I know, right?
So we actually don't know where, how the corneous beta proteins, feather proteins, we don't
hmm
know how those proteins evolved. There's one hypothesis that thinks that feathers evolved from avian scales. So these are
hmm
bird scales that you would see on a chicken foot.
Yeah.
Um, but there's another hypothesis that thinks that maybe feathers evolved from reptile scales. So the scales that you would see on a lizard. So we don't know from which uh, from which tissue of feathers evolved, and
Yeah.
we don't know how corneous beta proteins evolved.
So that's one of the things. Like, I mean, like you said, it's like it's kind of a love hate thing about science, right? Like, there's so many questions, and then almost every time you discover something, it just leads to more questions. But I also really, really appreciate when that is verbalized, because I feel like there's a lot of people who kind of have this idea of science as like science and scientists of like, they know everything, and it's like this elite group who just like they know everything and they know better. And it's like, no, actually, like, we know things, but there's a lot we have no idea. And like, and when people are open to saying that, like, no, I have literally no idea. We would love to figure that out someday. Like that, I appreciate that, because I think it's like, well, number one, it's an accurate representation of science, but two, I think it's good for people to know there's more to be answered, and might entice people to become scientists and things themselves.
Yeah,
But
and
yeah.
it certainly isn't some elite group by a long
Right.
shot.
Right.
I myself, I never thought that I could be a scientist. It is a lot of hard work, but it does not require you to be something that you think is exceptional. A lot of times you think,
hmm
oh, that person is exceptional, but no, not me. And it's not the case. It's a lot of small tasks that add up to big findings and big rewards.
Yeah. Yeah. It's yeah, it's a group, it's the world's biggest group project,
Yes, yes,
essentially.
yeah, you're right.
That spans thousands of years, and
Yeah.
it's quite complicated. Yeah, I'll say, like, when I saw you pop up on Twitter, and I was like, no way. Really? Like, and I don't mean that as like I didn't think that, because I want to be careful how I say this, because I don't want you to take it the wrong way. Um,
No,
I I just didn't expect it.
yeah,
I didn't expect it.
it's.
Um,
I feel
I mean,
like
I
if
didn't
it
expect where
was
I
to
am
be
either.
like
You know,
high school's most unlikely person to be a scientist, I would definitely raise my hand and say, yes, that's me. Yeah, so I would equally be shocked.
I
Um,
I was like both blown away and so impressed, and then I was like, yeah, it was just cool. It was really cool. Um, and I'm gonna be super super excited, because sometimes when I'm like doing these, my cord is tangled. Sometimes when I'm doing these conversations and stuff, I'm like, I wonder if there's a way I could incorporate. Like, I just was talking to an octopus biologist a couple of weeks ago, and she told me quite possibly one of the most mind blowing things I've ever heard in terms of biology. And I immediately was like, how do I incorporate this into teaching high school? Because, and I guess I'll just explain really quick, because that's kind of, I can't just put that out there and then not say
no,
anything.
you can't.
Um, okay, so transcription and translation of DNA into proteins. We typically think of the sequence as we have our DNA, it has to be opened and then transcribed and then it's translated into a protein. But at no point with the exception of like mutations and things, at no point is the code itself changed. It's just DNA and then it's made into a protein through a chemical process. Apparently, in cephalopods, they have so I'm going to save us a little bit of time. I explained it to Tiffany, but I just did a whole episode with Meg Menlin, octopus biologist, where we touch on this topic. So you know what? Go give that one. Go listen to that one if you want the scoop on the RNA editing. Okay. Thank you. And I'm
Oh
like,
my God.
no way. And I'm like, so immediately I'm
Oh,
like, okay, how do I incorporate this without causing confusion? But I've got to incorporate this because that's just so
oh
cool.
yeah. Whoa.
Um,
And see, this is, harping back to, your question of like, why does any of this matter? It's like,
mhm.
things like that. It's like, maybe you wouldn't think to look for something like this,
Yeah,
but you don't know what you don't know. And it's amazing how these things can take on a completely different relevance
right.
10
Yep.
years down the road. So we just don't fully understand. Of course we have implications of our research and hopes and desires of what it means and how it will add to our scientific knowledge, but you just can never really know what the implications of your research are, because 100 years from now, there's no telling, what
Right.
that'll mean and how that can change things.
Yeah. Yeah. Well, I mean, even in this kind of thing, like the amount that it bleeds into pop culture and
Yeah.
stuff is kind of mind blowing.
Yeah.
Um, and like how pop culture will adapt and stuff based on the new findings and things. It's
Yeah.
and I know like there's some people who are like my dinosaurs don't have feathers, and I'm like, I hate to break it to you, it's not your choice. Like this is what it
Yeah.
was, and we
Yeah.
gotta adjust our understanding of them. But there's something that's really beautiful about that to me as far as like understanding things in their own context.
Yeah.
Yeah, you start to look at things within the context of the organism itself, and it opens up so many more doors and so much more understanding. And I feel like this is just like it's a way of doing that, but through evolutionary history and time itself.
Yeah.
It's poetic. It's really cool.
Yeah. 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 Definitely.
That was my rambling. I got excited.
No,
Um,
I love it.
yeah. Now, we are just about done with this episode, but I've got one more bonus fact to share with you. And it's all because toward the end of our conversation, we were just trying to make sure we had covered all the things that we had planned to talk about. And she brought up something. And, well, you know what? I'll just let you hear it.
And then
Shoot.
if you wanted to, there was just the other question of how
Yeah.
do we age fossils? And I was just going to touch on that.
Oh, sure. Yep.
Oh, sorry. No, you were meaning how do we date fossils? Okay. Yeah, yeah. No, I just realized that. There, yeah, okay. Maybe this isn't
Wait, is there another meaning that I'm
well,
reading? Because now I'm like,
there
whoa, wait.
actually, yeah, there could be another meaning to that. So okay. I'm what you call within the discipline of paleontology,
Mm-hmm.
I'm what you call a taphonomist.
Mm-hmm.
So taphonomy is the study of fossilization processes.
Mm-hmm.
So fossilization is an incredibly complex process. You're talking about different geological events that have been happening over hundreds of millions of years. And we need to know how these processes can affect fossils. So if we dig up a fossil tomorrow, we need to have a good understanding of its history and whether or not it was buried very deep, because as something gets buried deeper, it undergoes more, it undergoes higher temperatures.
Mm-hmm.
So that is, of course, going to affect the preservation of your fossil. There's also things that happen where pore waters can come through, and you have oxygen in the water, and then that oxidizes your fossil. So you could have effects of oxidation in your fossil. There's
Hmm.
also minerals in that are carried in water. And then you can actually get some of your tissues in the fossil replaced by minerals. So there's all sorts of different processes that are going on that help create a fossil. And some of these processes will take information away from your fossil, and other processes might add information to your fossil.
Sure. Yeah.
So it's really important that when we're looking at fossils, that we're interpreting what we're seeing with this perspective of
Hmm.
understanding its history. And so one way that we help make sure that our interpretations of fossils are accurate is by doing taphonomic experiments. So this means that a lot of times I'm in the lab performing experiments on modern animals. So
Hmm.
it gets smelly very fast. And I, I basically use these tissues like modern bird feathers, and I'll I'll perform different experiments on them to see how different fossilization processes can alter the feather. So, for example, I might figure out the temperatures that might be relevant
Hmm.
to fossil feather preservation and bake my feathers in an oven and so I analyze it before treating it, and then I analyze it after treating it and see how some of these processes like temperature and oxidation and water, how all of these things can affect how a fossil is preserved. Um, and this will then help me because I can compile that into a data set, and then I can compare that data set to the data from fossil feathers. And then it helps inform my interpretation so that I'm not misinterpreting anything that might just be a reflection of the fossilization process itself and not actually reflective of how that feather was back
Sure.
155 million years ago or so.
Yeah.
So,
Yeah.
does that make sense?
It does. Yeah.
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 So in effect, we're kind of artificially aging fossils in the lab so
Hmm.
that we can then use that data to compare it to fossil data.
That is that. I did not realize like, okay, so now I'm gonna have to understand that there's a difference in those in those phrasings. But that I had no idea. I had no
Mm,
idea. And so
yeah.
how do you say, just so I want to get the spelling and stuff right too when I am working on this later. So you would specifically be identified as what?
A taphonomist. So it's
Taphonomist.
Taphonomist. And so the
Okay.
study of fossilization is the field of taphonomy. T
Hmm.
A F C A T A P H O N O M Y. Yeah.
Okay.
Yeah, and it is important because you could be looking at a fossil and let's say you interpret a structure as being skin,
Yeah.
but then actually when you look at um, let's say um, something that's not skin, so muscle. Okay, let's say muscle, and then you look at how that degrades, and then you think, wait, when this muscle is degrading, it's looking like skin. No, that's
Hmm.
not the case, but just as an example that it's very important because some things can degrade to look like
Yeah.
other things.
Yeah. Yeah. No, I can totally understand that. Huh. That learn something new every day. I'll take it like that.
Um, I hope I didn't. I hope this was all right, Devin. I
Yeah. Yeah. For sure. Yeah. I just this was well, I mean, as we can both attest, I think I'm thinking back. Gosh, how long has it been? 15 years?
Oh my
About
God,
15
right?
years,
Yeah.
Yeah, I'm feeling old. Yeah, I've got like kids, my hair's thinning. It's like, gosh, 15 years. It's, thinking back 15 years, I don't think, life is unpredictable. I never in a million years would have imagined that someday I'd be sitting here in my very old new house um, in Minnesota with two kids talking to you in Ireland about taphonomy and fossils and like I never like I could have that. It feels like a Mad Lib almost. Like I just wrote random words down on a paper somewhere, and then that's what happened.
Wasn't it?
So
Yeah.
yeah, that's just it's cool. It's very very cool. It's a
Yeah.
very odd but good experience.
Yeah,
And I really
absolutely.
enjoyed talking to you and catching up a bit, and that you were willing to take the time to do this.
Oh,
Um,
so willing, Devin. Like, I'm
yeah.
so appreciative. I'm so pleased that you asked me. And yeah, I hope it doesn't disappoint your listeners anyway.
And here we
No,
are. The end of our conversation, the end of today's episode. And hopefully the start of a lot of curiosity chasing for those of you listening. I mean, we talked about a lot of different things today. The fossils, the feathers, the particle accelerators, all of that stuff is amazing. It is mind-blowing. It is just the coolest. I mean, how could you not think it's the coolest? But if there's anything I really want you all to take away from today's episode, it's actually from earlier in the conversation when Tiffany was talking about her path, her advice, and we were talking about our educational experiences. And it's that we all have visions of what we want the future to be. We all have goals, we all have aspirations, some of them clearer than others. And of course, that is great. That's amazing. But always leave yourself a little bit of room, because you really never know where exactly it is that life is going to take you. You don't know which paths you will diverge from others on. You don't know when you may converge with them again. You don't know until you live it. How's the saying go? Life happens when we're making plans. So please, as you look forward to the future, as you make plans, as you think about what is next for you, leave a little bit of room for curiosity, for chasing that curiosity, for learning, for trying something new, for taking on challenges you never would have seen yourself taking on, for doing the unexpected, for asking all of the questions. And remember, wander often and wonder always. If you would like to support the making of this show, you can do that at patreon.com/thewildlife for as little as $1 a month. Now, I'll be honest, I would like to be more active on Patreon than I am. Things, life, parenting, teaching, high school, making this, and trying to stay sane through it all means that posting regularly on Patreon is not my strongest of suits. But know that any contribution really does help. Running a podcast can be kind of tricky, kind of expensive, especially when you have the interview platforms, the website hosting platforms, the subscriptions for music and sound effects and editing software, and holy smokes, it can be a lot. But I keep doing it because I love it. And of course, you owe me nothing, but any help genuinely is appreciated. For more information on several of the things that we discussed in today's episode, check out the episode notes, where you will also find Tiffany's social media information if you'd like to give her a follow. Because again, like I said, she has been just pumping them out, like awesome papers and discoveries and just some cool stuff. Cool stuff. She's a cool person, but you know that now. And the last thing, I've been considering something. Revising the Wildlife Book Club, in part because I have a lot of interviews coming up related to upcoming books. So I'm reading quite a bit. Some of them are pre-release, not quite out yet. Some of them have been out for a while. So be on the lookout for more information about that, because yeah, I would love to get that going again, reading something in common with all of you listening. And with that, take care of yourselves, be yourself, and peace out, Rainbow Trout. Thanks for listening.